Hello, everyone, and good morning. Thank you for joining us today. My name is Michael Yoon, and I'm a technical application scientist at IM Technology. In today's webinar, we will be introducing the latest advancements in intravital microscopy and how this technology is being applied in drug development research, particularly in the field of cancer immunotherapy. We hope this session provides you with valuable insights that can support and inspire your own research. Before we begin the main presentation, I'd like to introduce today's speaker, Jenny Ohm. Jenny is also a technical application scientist here at IBM Technology. She leads our CR research service and has been developing a variety of preclinical applications based on intravital microscopy. In her talk today, Jenny will highlight how intravital microscopy and IBM technology's unique capabilities can serve as powerful tools in drug development. She will also present several case studies focusing on cancer immunotherapy research using these approaches. If you have any question during the webinar, please feel free to submit them through the Q&A box. We will address them during the Q&A session at the end. Now, without further ado, let's welcome Jenny to begin the presentation.
Thank you, Michael, for the introduction. And hello and good morning, everybody. I'm Jenny Ohm, Technical Application Specialist at IBM Technology. Thank you all for joining today's webinar on the visualization of immune cell biodistribution in cancer cells for cancer immunotherapy research. In this session, I will introduce how IBM's intravital microscopy can be applied in the development of immuno-oncology therapeutics, and will also introduce several application cases in detail. At the end of the session, I'll present a brief virtual demonstration to showcase how our intravital microscope is used in actual experiments and the overall imaging process from start to finish. We will then finally address any questions you have during our Q&A session. Now, without further ado, let's begin with the webinar. As many of you may already know, the development of a new drug typically takes between 10 to 15 years and requires an investment cost of approximately 1 to 2 billion US dollars. Despite this enormous commitment of time and resources, unfortunately, 80 and 90% of drug candidates fail during the clinical stages. In fact, only about 5% of drug development projects ultimately succeed in reaching the market. The drug development process is granularly divided into 4 main steps. First is target identification and lead discovery. Second is preclinical testing. Third, clinical trials. And finally, regulatory approval and post-marketing surveillance. Among all the new drugs that are put into development, around 60% of the drugs that fail when transitioning from the preclinical phase to the clinical phase. There are many reasons for these failures, but a major factor would lie in the discrepancy between the preclinical and clinical results. This is largely due to the limitations of conventional preclinical approaches, such as in vitro assays and endpoint-based methods, which often fail to accurately reflect the drug's behavior and efficiency in living organisms. As a result, the drugs may not reach the intended tissues effectively, it may not act precisely on the desired targets, or it may cause unexpected side effects, all leading to clinical failure. Ultimately, to address these challenges, we need new technologies that can allow us to precisely observe and understand how drugs behave in real living biological systems and organisms. One powerful technology that can help overcome these limitations is intravital microscopy. Intravital microscopy is a high-resolution imaging technique that allows for real-time observation of cellular and tissue-level dynamics within the living organism. Traditional imaging methods, such as in vitro or ex vivo imaging approaches, typically rely on fixed tissue slides or cultured cells. As a result, they lack the ability to capture real-time biological and physiological changes and do not fully reflect the complexity of the in vivo environment. While modalities such as PET or MRI do offer in vivo imaging capabilities, they primarily provide insights at the organ or tissue level and lack the spatial resolution required to observe individual cells in action. In contrast, intravital microscopy enables the real-time tracking of drug behavior, biological responses, and disease progression, etc., at the single cell level in living animals. It is a powerful tool for visualizing various biological phenomena, including drug delivery and distribution, cell-to-cell interactions, immune responses, and so on, all with high spatial and temporal resolution. As mentioned before earlier, one of the most traditional methods used to analyze drug responses in the drug development process is the endpoint method. This approach involves sacrificing animals at specific time points to collect tissue samples for histological analysis. However, this method has significant limitations as it cannot adequately capture the variability of drug responses between individual subjects and falls short in providing a precise understanding of drug distribution or mechanisms of action within the organism or animal. As a result, it becomes difficult to accurately evaluate a drug's efficiency, increasing the likelihood of failure in clinical trials. In contrast, intravital microscopy allows for real-time longitudinal tracking of drug responses within the same individual subject over an extended period of time. This makes it possible to precisely determine how effectively the drug reaches the target tissue, determines through which pathways the drug acts, and whether the drug causes off-target toxicity in non-target tissues. Furthermore, because repeated measurements can be taken from the same subject, intravital microscopy can significantly reduce the number of animals required for experimentation, leading to savings in both time and cost. To acquire a more precise, reliable, and replicable data during in vivo imaging with intravital microscopy, IVIN technology has established and optimized fluorescent labeling protocol specifically designed for in vivo applications. This involves conjugating fluorescent dyes to antibodies and administering them via intravenous injection. Importantly, the injection dose is precisely controlled at 25 to 33 milligrams per dose, which minimizes variability caused by dosing differences and ensures both accuracy and reproducibility of the experiment. Moreover, imaging can begin immediately after injection, significantly enhancing experimental efficiency and reducing time overall. This optimized labeling protocol plays a key role in ensuring high-quality data acquisition and maximizing workflow productivity. Now let's take a closer look at the distinct advantages of IVIN technology's intravital microscopy and how it stands out from other imaging modalities through various applications. To advance research in fields such as immunology and drug delivery, it's essential to understand how and why biological processes occur within the living body at the cellular level. IVIN technology's intravital microscope enables dynamic, real-time visualization of cellular responses and disease progression in vivo. This capability provides critical insights that help guide the development of effective therapeutic strategies. Furthermore, by using live animal models, researchers can track the pharmacokinetics and pharmacodynamics of drugs over time or in real time, which is a powerful advantage in preclinical development. These functions also support the generation of highly reliable data that meets the regulatory standards required for drug development. As shown in the examples here, on the left, we see pulmonary vascular blockage in the lungs induced by inflammation in a sepsis model. And on the right, we can see that the distribution of exosomes and anti-cancer agents, as well as the dynamics and migration of transplanted stem cells, has been imaged and tracked. In addition to its imaging capabilities, IVIN Technologies' intravital microscope is provided as an all-in-one system for easy usage. This all-in-one design system simplifies installation and maintenance, and it eliminates the need for a dark room, allowing users to focus entirely on imaging and performing experiments without constraints from space or the surrounding environment. The system incorporates several advanced features that ensure exceptional in vivo imaging performance, including a high-speed polygon scanner, a real-time temperature monitoring system, motion compensation technology, and a soon-to-be-launched AI-powered noise reduction software. The motion compensation feature corrects for organ movement, enabling sharp and accurate imaging, even in organs with significant motion, such as the heart and the lung. The real-time temperature monitoring system allows researchers to track and adjust body temperature during long imaging sessions, ensuring compliance with animal welfare standards and minimizing physiological variation for more reliable data. Lastly, our upcoming AI-based noise reduction software will enable true quantitative analysis by effectively removing noise while preserving the original signal, ensuring clean and precise data sets for further data analysis. IVIN Technologies' intravital microscope is designed not only for short-term, but also for long-term imaging, ensuring stability throughout extended experiments. To support this, we provide a wide range of customizable imaging windows and chambers that facilitate stable, long-duration imaging across various organs. These chambers also enable real-time pharmacokinetics and pharmacodynamics analysis by allowing direct visualization of drug movement and distribution within the body over time. The current slide shows examples of some of the most widely used window and chamber designs of IVIN technology. If your research focuses on a specific organ or requires specialized conditions, we are happy to assist with the custom development and optimization of imaging window chambers tailored to your experimental needs. If you are interested or have any inquiries regarding this, please feel free to reach out to us to discuss further on how we can support your research goals. By using IVIN Technologies' intravital microscope, we can visualize various organs in real time in living animals. This enables direct observation of a wide range of biological processes, such as immune responses occurring in vivo, tumor progression and metastasis, drug delivery and mechanism of action, and more. Let's take a look at a few representative examples. Firstly, depending on the drug formulation, we can observe differences in immune activation induced by various drugs. We can also evaluate therapeutic efficiency during cancer metastasis, visualize fibrotic targets for non-alcoholic steatohepatitis therapeutics, and assess the delivery and efficiency of various drug formulations. These examples illustrate how intravital imaging offers powerful and mechanistic insights across many therapeutic areas, from oncology to immunology and fibrosis. Now, I'd like to walk you through a few experimental cases that were conducted by using IVIN Technologies' intravital microscope. These examples will give you a deeper look into how the system is applied in actual research settings and how it can provide critical insights across different stages of drug development. The core of immuno-oncology therapeutics lies in stimulating immune cell activation, which enables the effective elimination of cancer cells. Therefore, directly observing how immune cells respond and move in the tumor microenvironment is crucial to understanding the mechanism of action and therapeutic efficiency of these treatments. In the following experiments, we tracked the infiltration and migration of immune cells in real time, enabling us to observe temporal and spatial changes in the immune response. As mentioned earlier, in order to enhance the efficiency of immuno-oncology therapies, it is crucial to understand how immune cells infiltrate the tumor, as well as when and how they become activated. Immune cells play a key role in regulating immune responses within the tumor microenvironment. So precisely tracking their behavior provides valuable insights into the mechanism of action of immuno-oncology agents. This also helps in designing new therapeutic strategies. With this background in mind, the first experiment I'd like to share involves real-time imaging of immune cell infiltration in a synergic mouse tumor model. In this experiment, immune cells were visualized using a transgenic mouse, and the dorsal skin fold chamber was utilized in order to observe the infiltration of myeloid cells into the tumor over an extended period of time. Two different cancer cell lines were injected into two separate groups of mice, and imaging was carried out at multiple time points, 2 hours post-injection, and then from day 1 to day 9 post-injection, in order to capture the temporal patterns of myeloid cell infiltration and monitor their activation. If we now look at the results in the first experimental group, where melanoma cells were injected to create the tumor model, the results showed that at the 2-hour post-injection time point, immune cells were not visibly detected, and there was no apparent infiltration of immune cells into the tumor. However, by day 1, immune cells began to infiltrate the tumor and over time gradually further infiltrated into the tumor as well. By day 9, this infiltration was most pronounced. So in conclusion, the activation of immune cells by the tumor cells began at around day 1, and the significant infiltration of the immune cells into the tumor started on day 2. In the second experimental group, colorectal carcinoma cells were injected to create the model, and much like group 1, no immune cell infiltration was observed at the 2-hour post-injection time point, with immune cells first appearing within the tumor microenvironment on day 1 post-injection. As time progressed, the immune cells increasingly invaded the tumor, but unlike in the first group, the immune cells also spread beyond the tumor margins into the surrounding tissue. In conclusion, in the second group, immune cell activation began on day 1. Significant immune cell infiltration was evident by day 3, and notably, the extent of infiltration was much broader and wider in the second group compared to the first group. As one of the most critical points in immuno-oncology research, it's essential to directly confirm how effectively an administered drug activates immune cells in vivo and whether these cells migrate to and act within the tumor. In particular, T-cell infiltration into tumors is considered a key predictor of immunotherapy responsiveness, making a visual assessment method for drug action absolutely necessary. Taking this into account, the next experiment was designed to evaluate the in vivo efficiency of an immunostimulatory agent. The slide you're viewing shows data collected before administration of the drug compound. In this study, wild-type mice were first implanted with a dorsal skin fold chamber and then inoculated with melanoma cancer cell line. The drug compound was then administered subcutaneously, and imaging was performed on day 4 of post-injection. Here, if we look at the results after post-injection of the drug, we can see that at both spot 1 and spot 2 regions, where the cancer cells are clustered, and we could also observe that the T cells were localized within the vasculature located near and around the cancer cells. Upon closer examination, as shown on the left, we were able to confirm that T cells were actively migrating towards the tumor cells. On the right, we observe the distribution of T cells within the blood vessels surrounding the tumor, which provides strong evidence that the administered drug effectively activated immune cells in vivo. This also serves as an important indication of the drug's ability to stimulate immune responses within the tumor microenvironment. Now, the previous results shown are insightful pieces of data into understanding how immune responses change in the tumor microenvironment. But it's difficult to assess the efficiency of immuno-oncology therapies based solely on changes in cell counts. Instead, direct visualization of how immune cells move and interact within the tumor microenvironment is essential. From here, using real-time imaging data of in vivo immune cell dynamics and distribution, I'll demonstrate how this high-resolution intravital microscopy technology can be applied to advance immuno-oncology research. Understanding how immune cells traffic or migrate throughout lymphoid tissues before reaching the tumor is critical for optimizing immuno-oncology therapies. In this study, high-resolution intravital imaging was used to visualize lymphocyte trafficking, specifically T-cell migration within the lymph node. This study specifically focused on the HEV, or high endothelial vannule region, which is the key gateway through which T cells, B cells, and NK cells enter and exit the lymph node. Within this microenvironment, lymphocytes undergo a well-defined sequence of steps: rolling along the endothelium, firm adhesion, and then transendothelial migration into the lymph node parenchyma. In the video in this slide, you can see activated T cells engaging in this process. They roll along the HEV, adhere to the endothelial surface, and then transmigrate through the vessel wall into the lymph node. This directly demonstrates the dynamic behavior of T cells during the early stages of immune response. Building up on the previous data, this next video captures what happens after T cells have traversed the HEV. You can now see them navigating through the surrounding fibroblastic reticular cell, or FRC network, which surrounds the HEV structure. This allows us to precisely observe how activated T cells migrate through the lymph node's complex stromal framework following transendothelial migration, providing deeper insight into the cellular pathways that underlie the initiation of an immune response. To accurately evaluate the therapeutic efficiency of immune checkpoint inhibitors, is not only important to determine the presence of immune cells, but also to assess whether these cells successfully infiltrate the tumor, proliferate, remain motile, and actively perform their immune functions within the tumor microenvironment. Following the previous imaging data, which showed how T cells become activated and migrate through complex structures within the lymph node, I'd now like to introduce an extended study that visualizes how these activated T cells, also known as tumor-infiltrating lymphocytes, behave inside the tumor microenvironment. Specifically, we examined how effectively these tumor-infiltrating lymphocytes, or TILs, infiltrate into the tumor and how functionally active they are once inside that microenvironment. In this experiment, we used a breast cancer mouse model, and using intravital microscopy, we observed TIL infiltration patterns and dynamics over time on days 1, 3, 8, and 11 after tumor cell implantation. As a result, we were able to clearly observe the proliferation patterns and activation status of TILs over time. Notably, by day 11, TIL activation and proliferation were at their peak, indicating the most robust immune response within the tumor microenvironment at that time point. Additionally, the cell tracking data shown below quantitatively analyzed the migratory behavior of TILs over the same time course. On day 11, TILs exhibited the longest travel distances and the highest migration speeds, suggesting that their functional activity was also at its highest during this period. Additionally, to analyze how the degree of TIL activation and infiltration differs between the interior and exterior of the tumor, we tracked and monitored both regions separately. As a result, a higher number of TILs were observed within the tumor compared to the outside of the tumor. Track-on data and quantification graphs further confirmed that TILs inside the tumor traveled longer distances and exhibited faster migration speeds than those outside the tumor. These findings suggest that immune cells do not simply remain around the tumor, but actively penetrate deep into the tumor microenvironment, where they move dynamically and perform their functions. This behavior serves as a critical indicator for evaluating the efficiency of immune checkpoint therapies. Notably, the functional activation of TILs was significantly higher inside the tumor than outside the tumor, indicating that the extent to which immune therapies can affect the inner tumor microenvironment is a key predictor of therapeutic response. Therefore, to maximize the effectiveness of immune therapies, it's essential to have systems that can precisely evaluate TIL infiltration and activation within the tumor. As demonstrated in this study, real-time imaging-based analysis serves as a powerful tool to achieve this goal. Lastly, one of the key strategies to enhance treatment response in immuno-oncology research is to maximize the synergistic effects by combining immune checkpoint inhibitors with existing therapies. Radiation therapy, in particular, has garnered attention for its potential synergy with immunotherapy. Not only does it eliminate localized tumors, but it can also expose tumor antigens, thereby stimulating immune responses. The experiment I'm about to introduce was designed to explore the potential synergy between immunotherapy and radiation therapy. Specifically, it aimed to visualize how these two treatments influence immune cell activation and tumor response in vivo. In this study, a breast cancer model was established by injecting a breast cancer cell line into the flank region of nude mice. The animals were then divided into two groups. One group received immunotherapy alone, while the other received a combination of both immunotherapy and radiation therapy. Intervital imaging was performed on days 1, 3, 7, and 10 after the start of treatment to monitor tumor response, allowing for a direct comparison of therapeutic effects between the two groups. As a result, a notable increase in T-cell proliferation was observed in the group that received both immunotherapy and radiation therapy, particularly in the later stages of the experiment. By day 7, T-cell activation and expansion were most prominent in the combination therapy group, highlighting the potential for a synergistic effect between the two treatments, a key point in evaluating combination strategies. By day 10, while the number of T cells had decreased, dendritic cells were observed in the tumor microenvironment. This suggests dynamic shifts in the immune landscape and interactions between immune cell types, potentially indicating a transition into the next phase of the immune response. In addition, quantitative analysis revealed a significant reduction in tumor size. Notably, tumor shrinkage was observed as early as day 7, and when comparing the two experimental groups, a clear reduction in tumor size was evident in the group that received combination therapy with radiation therapy, which indicates the influence of the combined therapy on tumor size reduction. In conclusion, these results demonstrate that combining immunotherapy with radiation therapy can enhance immune activation and ultimately generate a synergistic effect in tumor elimination. So far, we've walked through various application data sets that highlight how IVM Technologies intervital microscopy system can be utilized in immuno-oncology research. To wrap up, I'd like to briefly demonstrate how efficiency studies using IVM Technologies intervital microscope are actually conducted, giving you a quick look into the experimental process of evaluating a drug using this technology. So in this demonstration, we conducted an experiment to evaluate whether or not the drug could activate immune cells within the liver and to observe how the distribution of these immune cells changes in response to the treatment. So first, let me briefly introduce the configuration of IVM Technologies intervital microscopy system. So what you're looking here is our IVM CMS3 main unit, and below the unit, we have our confocal laser, and we have our 2-photo laser below that, and at the very bottom, we have our power supply installed. For imaging, the mouse is mounted on a stage, which includes a heating plate with an integrated temperature control system connected directly to the main body of the system. Additionally, included in the setup is the temperature monitoring system. So the temperature monitoring system allows the user to maintain and adjust the body temperature of the mouse throughout the experiment. And we also have a jog dial that is installed with the system. And the jog dial provides precise control of the stage position, allowing the user to move it to the desired imaging location. Next, in order to perform long-term imaging and track drug distribution, we surgically implant an abdominal imaging window chamber into the mouse. This chamber enables long-term in vivo imaging and allows real-time visualization of how the drug distributes throughout the body. What you're seeing here is the overall process of that surgical procedure of implanting the abdominal imaging window. Once the procedure is complete, we mount the mouse onto the intervital microscopy system for imaging. Next, I'll show you how we use the IVM engine for imaging and the IVM Studio for post-processing. So on the left side is the overall interface of the engine, and you can change the values accordingly to how you want to perform your imaging. Then you use the jog dial to move the stage to identify a meaningful reference region for imaging and perform your imaging process accordingly. Once you have acquired the data, you can then use the IVM Studio, as you can see in the current video, for post-processing, additional editing, and further 3D analysis. So this is the results of the previous video I have shown you. At the 24 hours post-injection time point, we conclusively observed a marked increase in drug distribution compared to immediately injecting the drug, suggesting that the circulating compound or drug had diffused out of the vasculature of the liver and adhered to the perivascular tissue in the liver. Importantly, we also saw a rapid rise in immune cell numbers, forming distinct clusters at this time point, strongly indicating that the administered drug effectively activated these cells in vivo. Again, if we take a closer look immediately after drug administration, some immune cells were observed. However, by 24 hours, there was a rapid proliferation of these immune cells, forming distinct clusters. When comparing the signals of T cells and the drug, the proliferation of T cells was much more prominently increased at the 24-hour time point. In addition to the lymph node, dorsal skin, and the liver that I've previously shown, our system also allows high-resolution in vivo imaging of various organs in live mice, including the brain, lungs, spleen, and heart. Furthermore, IVM technology not only provides an all-in-one system capable of high-quality in vivo imaging, but we also offer CRO research services based on researchers' samples. This enables more efficient and specialized research support to help facilitate your studies. The screen you're currently looking at showcases some of our customers in which we provided our CRO research services. We have worked with a range of companies, research institutes, and universities offering both short-term and long-term imaging CRO services. Now let me briefly walk you through the process of how our CRO research services are carried out. The CRO research services provided by IVM Technology follow the steps outlined below. First, we begin with a consulting phase where we have a detailed discussion on the experimental scheme. Following this, we have a feasibility test or a pre-test that is conducted to evaluate whether or not the proposed experimental design can be effectively executed. Once the pre-test is passed, we move on to the main experiment phase, where in vivo imaging and data collection takes place. Finally, post-processing and analysis are performed, and the results are compiled into a final report. For more details regarding our CRO services, please feel free to contact us via the email address below at information@ivmtech.com. So thank you everybody for joining us today for the webinar. Over the course of the session, we explained why intravital microscopy is vital in the drug development process and how it can be applied. We also highlighted IVM Technology's unique intravital microscopy system and shared some real-world application cases acquired with the system. We sincerely appreciate your time and attention, and that concludes my presentation. Thank you.
Thank you, Jenny, for insightful presentation. We will now begin our Q&A session and answer some questions. The first question is, what are the animal species used in intravital microscopy studies?
So the animals that were used in the studies I showed you in my presentation were mostly mice, but with intravital microscopy, not only mice can be used, but rats, guinea pigs, or any other animal that you're currently using for your studies can be applied with intravital microscopy.
Second question is, if you are imaging on 2 consecutive days, do you need to inject your fluorescent antibodies before each imaging session? And how long can they be detected following injections? Also, these injected via the tail vein or different IV site?
So they are injected intravenously, typically through the tail vein. But if tail vein injection is difficult, you can also inject it through the rectal orbital vein. And if you're imaging on 2 consecutive days, you would typically need to inject one more time once the 2 days has passed, since the antibodies that are conjugated with the dyes last about 2 days. So I think that could answer your second question, which is how long can they be detected following injections? Following injections, it can be detected right away, but we do recommend to wait about 15 to 20 minutes after injection before you start your imaging.
And next question is, what is the cost of the magic system?
So the cost varies according to the components, but the typical range would be from 400,000 USD to 600,000 USD. But if you give us an email to our address, we will tell you more in detail about the basic cost of the system.
And next question is, with the IVM protocol, are you able to conjugate fluorescent dye to materials other than antibodies?
Yes, you are definitely able to conjugate the dyes with other materials. And At Iving Technology, we also consult and help you with custom labeling, which is where we can conjugate fluorescent dyes to your research samples. So if you're maybe interested in inquiring about custom labeling, or if your research sample is able to be conjugated with our protocol, again, you can give us an email at information@ivingtech.com.
And this is the last question. In the long-term experiments where tumor cells were observed over several days, how did you manage to image the same location consistently?
So typically when we look for the same spot, we look for blood vessels, as blood vessels typically do not change over time, or we target immobilized cells. And we are optimizing different methods physically and on our chambers to let the user find the same spot every time they image. But currently we recommend and conduct finding the same spot using blood vessels and immobilized cells.
Thank you everyone for all your questions. If you have any more questions or if we couldn't address your inquiry during the Q&A session, please feel free to reach out to us at information@ibm.com, and the webinar recording will soon be available on our YouTube channel. At IBM Technology, we offer both on-site demos starting from a couple of days to weeks, and we also offer virtual demos through Zoom. We can also test your samples through our CRO research service and provide training for new animal surgery models for long-term or terminal intravital imaging. We appreciate your interest in our webinar today. We thank you for your time and look forward to seeing you again soon. Thank you.